Cetaceans, whales and dolphins. These animals have captivated me since I was very little, maybe 6 or 7 years old, I think. And though I can't get enough of the biology on this fascinating group of mammals in general, it was their evolutionary history that inspired the most wonder in me as a child. I've always liked the water and swimming, so I think a part of me responded very emotionally to the concept of adapting to a completely aquatic lifestyle from a terrestrial one. As a group, marine mammals, pinnipeds and sirenians included, highlight the process of evolution very well. All animal life on Earth started off aquatic, of course, and mammals as a class represent a very specific set of adaptations and strategies for surviving on land. And then we have members of this terrestrially specialized group adopting more aquatic lifestyles again. Evolution doesn't go backwards, at least not in any major sense, so what we have is a modification of the basic mammalian life history plan for aquatic living, not a reversion back to a fish-like condition. Millions of years cannot be undone. Time only moves forward, and that is the direction natural selection and descent with modification work in. This concept is obvious, and it makes common sense. The physiological extremes that the mammalian body plan has adjusted to in marine mammals typifies this necessary forward progression. Systematics as a field of study is the understanding of biological diversity through the reconstruction of evolutionary history or phylogeny. Its important contributions to biology in general are manifold. For one, systematics allows for the interpretation of the changing characteristics of organisms over time, their direction, frequency, and relationships with each other. Take as an example the co-occurrence of complex social behaviors with high brain cognitive capacity in multiple animal lineages, including primates and cetaceans. Systematics also enables hypotheses of adaptation to be tested. For example, did echolocation evolve once or many times among cetaceans? Conservation priorities can also be highlighted with systematic information. Losing the baiji or Chinese river dolphin to extinction, for example, meant the loss of a unique and old evolutionary lineage that represented an entire taxonomic family on its own. Systematics can allow for the prediction of characteristics on poorly known organisms based on their evolutionary relationships to well-known ones. For example, some species in the family Zephiidae, the beaked whales, have never had confirmed sightings at sea and are only known from washed-up remains. But based on the morphological evidence provided by those remains, we can predict that they have foraging behaviors similar to other better-known Zephyids. Finally, systematics provides the foundation for studying other biological patterns in an evolutionary context. Take, for example, the coevolution of hosts and parasites. Before jumping into the evolutionary history of cetaceans, let's first go over some basic systematics terminology. Characters are the features or attributes of an organism. They can be morphological, for example, the presence of external ear pinnae, or they can be molecular, such as the base pairs present at each nucleotide position of a gene. The different ways the character can manifest are called character states. In the case of external ear pinnae, this is either presence or absence. And in the case of DNA, each nucleotide position can have one of 4 character states corresponding to each of the 4 base pairs. Plesiomorphic is a term that refers to a character state that is ancestral. For example, the presence of hair on humans is an ancestral character state shared among all mammals, so this character state is plesiomorphic in humans. The coelacanth is a sarcopterygian that has many plesiomorphic character states and likely resembles the common ancestor we as humans share with it. By contrast, apomorphic refers to a character state that is derived. For example, baleen is an apomorphic trait in mysticetes. It's important to understand that only characters can be plesiomorphic or apomorphic. An organism or species as a whole cannot be primitive or modern. One may have the urge to call the coelacanth a primitive fish, but it's not. It's just as modern as you or I, since it's alive today. It just has many character states that are ancestral among sarcopterygians. It is a lineage that has not changed as much from the common ancestor it shares with lungfish and tetrapods like us, though this does make it a unique lineage with no close living relatives, and living coelacanths represent an entire subclass all on their own. An exaptation or co-option is an adaptation that takes on a function different from the one it had originally. For example, the forelimbs of terrestrial pinniped ancestors were originally adapted for walking on land, but these limbs were then co-opted to function for swimming. Cladistics is the term for the collective methodology used in systematics, and it operates under the basic principle that shared derived character states, or synapomorphies, are evidence of common ancestry. This history is represented in a cladogram or phylogenetic tree, an hypothesis for the evolutionary relationships among the organisms it includes. Branches represent taxonomic lineages, or clades, and nodes represent speciation events and signify common ancestors. Cladograms can be drawn so that branch lengths are proportional to evolutionary distance, and sometimes these can in turn be time-calibrated. When drawn as such, cladograms are usually referred to as phylograms. The direction of character evolution among your group of interest, or in-group, is inferred from outgroup comparison, where outgroups are representative species from lineages outside your in-group. A character state found in both your in-group and your out-group is thus likely an ancestral or plesiomorphic state in your group of interest. By contrast, a character state that is found in only your in-group organisms is thus likely a derived or apomorphic state in your group of interest. A monophyletic group of lineages contains a common ancestor and all of its descendants, and ideally speaking, taxonomic nomenclature should refer to monophyletic clades. The following groupings are also monophyletic. A paraphyletic group of lineages includes a common ancestor but only some of its descendants. E and F are the groups excluded in this case. A polyphyletic group of lineages are separated by more than two levels of common ancestry. Monophyletic groups are the most useful for taxonomic classification and for systematics since they are defined strictly by their ancestry. The clade can then be diagnosed, in other words, new organisms can be taxonomically classified, by the characters that are apomorphic in the group. But similarity in character states among a group of organisms can sometimes occur even if there is no shared common ancestry. A homology is a similarity in character states that results from common ancestry, for example, the basic skeletal plan of mammals. A homoplasy, by contrast, is a similarity in character states due to evolutionary convergence or reversal, not common ancestry. For example, the flying squirrel body plan has converged in two lineages of mammals, one of which is a placental mammal, the American flying squirrel, and the other of which is a marsupial, the Australian sugar glider. Distinguishing between homology and homoplasy depends on constructing phylogenetic trees based on as many different characters as possible. This way, collectively, the most likely evolutionary relationships will emerge,